I fed this slime mold an oat flake soaked in blue dye and then put it under the microscope. And let me show you the video that I got. Look at this. You can see the blue vesicles, these little like food pouches. Not just food, actually. Vesicles play an important role in all of cellular biology. They're just the packets that cells put stuff into. But these ones in specific, the blue ones that you can trace through throughout the cell as it's pumping cellular material around. These ones are the food vesicles. They're filled with little pieces and solubles from the oat flake that it's digesting over in that direction, and it's sending them throughout the whole cell. And the most interesting thing is you can see where they're accumulating. If we observe where they're accumulating in this one enormous cell with many, many nuclei, the cell has tens of thousands, if not hundreds of thousands of nuclei, all in one single cell. If we observe where the food is going, where it's being deposited, we actually get a clue about where these nuclei are. Are they distributed throughout the cell evenly with just diffusion, or is there a targeting mechanism that puts them somewhere? And the answer is yes. They're targeted to the leading front because this is where the most activity, especially protein synthesis, happens in the cell. And so that's where the food is being sent as well. It's also where the mitochondria are aggregating. I also think this video is such an interesting study of how cells color themselves and just how we perceive color. Because look at how relatively few packets of blue there are. There's very few blue dots here. But when we look at the picture, here's the picture again of the specimen we were looking at. We were looking right about here in that little tiny section. And look at how blue that looks. And of course, the cytoplasm we were seeing flowing was coming from this. And this looks very blue, actually. But when we saw it under the microscope, there was very few and very discrete packets of blue, but it was coming across as like this bluish green to our eyes just looking at it without any aid. Isn't that interesting? I think that means something for color science. Isn't that strange? And also the way that cells color themselves globally. This cell is usually bright yellow. Oh, I'll find a picture. So this is what it looks like with no dye at all. Well, no synthetic dye. Of course, the cell itself produces a bright yellow dye to achieve this. But without the dye, without this bright yellow dye, which actually also has somewhat discrete packets. Oh, I have another picture to show you. Okay, I'm sorry, this is not an amazing picture, but this is just a tiny little amoebal cell of this slime mold. And you can see the individual little packets, the vesicles of this. Now, this is a little different because this particular pigment is soluble in the membrane. So it actually exists in the membrane of the vesicles, and to a large extent, it will deposit into the membrane of the entire specimen. So it's a little bit of different story with this one. It's more evenly distributed. But it does still appear in many cases as these discrete packets. Anyway, it does not take that much pigment to produce extremely vibrant colors on this macroscopic scale. Microscopically, they can be in very discrete little packets, and there doesn't need to be a whole lot of them at all to really affect the color of this macroscopic creature. Isn't that interesting?